Kiro. I can't go look at test me, huh?
Yeah, only the screen so shared later. Tonight we'll start our program. We are really sorry for the inconvenience. And good evening, everyone. I am Dr. Lalremsanga, and I'll be the host for tonight. On behalf of Government Strategic College, I once again extend a very warm welcome to all the participants of the fifth night of one-week international webinar on science in the 21st century. I would like to thank our esteemed resource person for accepting our humble request in spite of his busy schedule. And to all our participants, thank you for sparing your valuable time with us, and please stay with us till the end of the program. Moving on to our program, our resource person for tonight needs no special introduction, especially in the field of life science. It's my pleasure to introduce tonight's resource person, Professor H. Head Department of Zoology, Mizoram University. Since we are running out of time, I will be highlighting only few of his CV. He has been awarded several prestigious awards from different organizations. To mention a few, he was awarded for the contribution towards discovery of 3 new species belonging to 3 different classes of vertebrates from Mizoram, and the Best Citizen Award 2019. And then he also received an International Herpetological Symposium HRS Grant Award 2021. And besides that, he is among the founder members of the biggest NGO in Mizoram, which we call BICON. And he won the Balipara Foundation Annual Award in 2015 during his president term for his contribution towards the nature conservation in Mizoram. And research around 140 publication and book chapters 21, book 2. And then under his research guidance, 2 MPhil and 1 PhD are awarded. And Sir is actively engaged in organizing different environmental awareness program among the local people and various institutions. And he's also a good campaigner for snakebite management, rescued hundreds of snakes where he survived 3 times after bitten severely by the cobras and viper, helping snakebite patients to get antivenom, and also collaborated with the premium serum Pune to supply adequate amount of anti-serum in a cheaper cost, especially for rural areas. And from his work, local people, we can say even the local people, the Mizos, are also changing their attitude towards the snake conservation. I can go on and on by reading Sir's CV. So now, may I now call upon our esteemed resource person, Professor H.D. Lalremsanga, to deliver his talk. Sir, please.
Good evening to all of you. Can you hear me?
Yes, sir, we can hear you.
Oh, you can see the screen, the slide.
Yes, sir. Full screen.
Is it moving?
No, it's
A
not,
full
sir.
screen low me.
Full screen low.
One more way on the full screen.
Oh, a full screen, yeah, sir, full screen.
Full screen em. A full screen em thì hiện.
Full screen le to la wa ma ro chu lang chiang thau mai.
One more về cái minimize chỉ hiện nào. Anh là one more. Ngồi ở gần hông thang nghe lóc à.
Sir ka alef pala mo ni oh. Lang hak le lok miyang.
lock so detect le thì to lo
Sir, mo sa team lock sharing akan mo present now your entire screen so katila suan chuminua I ing lau kha ilud dei don ne screen your entire screen ti kan ti chin ro e window ti ilu pau chuan ati lei thei lo win ka ria
entire screen ita tiro
oh
chuan
ti khan sharing ti kha ha khot lamo
oh
aron me me panga
ni china ha tiro lang mai ya
o khamialan nuwa po khan achang chuan annex thei lo thri no mo
oh hian hei hian
vela
entire screen chuan om ta mo engma
kan lam mumi elo ma se
lia entire screen ang chuan om ta mo emi ke ma ni Google Meet chok kami screen khan lang ta top ta ni chuan share ti
chuan
he blind ta ya
e khala khan screen kai han click lamo
oh
screen ka han click la chuan e share ti kharon nung chi mai thei
katika aron
screen ka han click la chuan share ti kharon polem
pol chuang lo
foma kwel
isoyom zia zo isoyom zia zo kami PPT on khani me tiro
a imo your entire screen at tiela na khan mo
ah
share your entire screen ti vel aron pop up anga tro chu an click anga chuan an noya share le cancel ti omna share kharon pol chiang
pol si lo chu chu nia
ama ka han click sinte englo share your entire screen tia I englo screen ron lang veka ka han click pot la
o pola
oh share ti khan share a pol chon share like
kana lang e
oh langamo oh mase ala full screen lo ta
full
ni
screen lawa de ka full screen ni na lang em
oh lang chi e
de kana tha mo
oh han next sinte an next thei lo thina changin
next time
ngor oh
next
next carbon oh next se atha etike
oh atan them can we start
Yes, sir.
okay tonight sorry for the delay and some technical problem this always happens in this webinar tonight my topic will be snakes venoms and antivenoms this will be in context to Mizoram and it will slowly I will enlarge in a broader area so snakes venoms and antivenoms so first we should know about this what is herpetology. It is the study of amphibians and reptiles. We know what are amphibians and reptiles, I hope. Then in East India there are big challenging because there is many unexplored area and many scientific studies in this regard and there are lots of mythology in connection to these amphibians and reptiles so we don't even know how many species of amphibians and reptiles we have in Northeast India. Mizoram also taxonomy and biosystematics It is only emerging here in this region. Information about their habits, habitats, and present status. So these all are still a question. So regarding their conservation actions also, every actions of ours in this regard will count. So whatever we start, whatever we conduct research on this herpetology, it is important. So on coming to reptiles, we know what are amphibians. They are frogs, Sicilians, salamanders, those which are devoid of skin, scales, and they are cold-blooded animal also. Then the other group, that is reptiles. These reptiles, they are long-breathing vertebrates, originally with 2 pairs of limbs, each with 5 clawed fingers and toes. Exceptional cases for snakes and glass lizard, limbless lizards. So this is a body covered with horny scales and plates and are devoid of hair and feathers. They are poikilothermas, that means all-blooded animal. So that's why they need extra heat in order to regulate their internal body metabolism. They are usually oviparous or ovoviviparous. That means they are egg-laying animals. Or those ovoviviparous, that means their eggs usually hatch inside the female womb. Then after that, it gave birth. So as the first two land animals, they reach peak of their evolutionary development during the Mesozoic era, a period known as the golden age of reptiles. I think we had seen this kind of dinosaurs in many movies. So the class Reptilia includes 10,000 species. Reptiles can be easily recognized by their horny or scaly skin. At present, the class Reptile has 4 orders. Number 1 is Squamata, that includes lizards and snakes. Then number 2 is Testudines, that contains tortoise and turtles, also known as chelonians. Then number 3, Crocodilia, that includes alligators, caimans, crocodiles, gharials, which are native to Indian subcontinent. Then Rhynchocephalia, it includes only 1 living species, the Tuatara. And earlier, they identified 2 species, Sphenodon punctata and Sphenodon gantheri. But this is the second one, Sphenodon gantheri. The status is still questioned. So on coming to the suborder, Serpentes, there can be variation of size from the smallest snake, that is Barbados test snake, Leptotyphlops carne, to this the largest snake in the world, that is anaconda. So there can be a variety of shape and size, coloration, then behavior, natural history also. Then in India, on coming to India, we have big 4 snakes of India. One is common crane, another one is Brazil viper, another one is saw-scale viper, then another one is spectacled cobra. So these are found, but these are found only in the mainland of India. They are not usually found in Northeast. They are not common. This one, it is found in the western part of Assam, and this one, it is also reported from Manipur. So these are known as big 4 snakes of India. Then among 3,956 species of snakes living on Earth, more than 278 species are known from India. So far, more than 110 species of snakes are recorded from the Northeast India. New species are keep on describing within this decade. More than 20 snake species in Northeast India are deadly venomous, excluding those big 4 snakes. So we started our work from examining roadkill, exploring different microhabitats during day and night time. So the most important part after collecting snakes, dead snake or live snake, is taxonomy. It depends on this morphological study. So scalation is important. Number of scale in the upper mouth and number of scale in the lower mouth, then number of scale upper eye, posterior, anterior, and behind the post-ocular scales also. All these scale number and the shape and size is important, as we have 32 teeth in human being, that is fixed, or we have 10 fingers like that. So this scalation, number of scales is important for identifying snakes, because same species usually are having same number of scales like that in this particular area. So this is the identification key for different scalation. So we established a museum in our department. Then from our work, we found these 3 different types of blind snakes. All these are fossil, and they are found in the soil and they used to come out during rainy season. All these are non-venomous. Many people, they believe that they are deadly venomous, but they don't even have mouth also. It is only pin size. And we have 2 types of python. The longest snake in the world, that is reticulated python, usually found in the southern part of Mizoram. Then this is the common one, Burmese rock python, Python bivetetas. So this is a recently collected reticulated python from southern part of Mizoram. Then coming to the family Colubridae. These snakes, they can poke the eye. There is a myth, and they are also used to tell that they are deadly venomous because the head is very prominent and very large like that and triangular. But they don't have fang and they don't have poison also or venom. So they are only mildly venomous because they can kill their own prey like a lizard, small lizards or rodents. So they used to give birth. They are ovoviviparous because after the egg get out in the female womb, then slowly they used to give birth for the neonate, okay, which is usually brown in color. Then within 4 or 5 months, the color will be turned into greenish. So this one is documented recently here in Aizawl. And another non-venomous like Habeas cassius, Herpetology, genera, Amphiosma stoletum. This Amphiosma stoletum is same species. This Champai pedophile. This one is from Champai lele, pedophile. These 2 types, they are different morph. One is Forma typica and the other one is Electrocistras. So we found this genetic variation among these 2. And not only between these, and there are difference in the, if we compare with other species, same species found in China and other countries like Myanmar and other parts of Northeast India. Then this is Herpetology. This is the first observation for Herpetology genera that feeds on frogs, that is Hydrophylax, Leptoglossa, and another snakes, Himalayan keelback. So this one we published in this Reptiles and Amphibian journal, and this is the first evidence for the maximum size of clutch size. Litter size is 7. That is maximum record. This is Habeas viningi, first record for the state, and this one is likely to be a new species found in the moorland. So this one we already communicated in the journal. I hope it will be coming out soon. Then we give the name for moorland, Herpetology moorland. Then another one, Blithia reticulata. Earlier, this one is Monotopic Genesis. That means under this Blithia genus, only one species was there, that is Blithia reticulata. That is why it is known as Monotopic Genus. Then recently, we found this, there is no studies on the biology, how many eggs it can lay, and during which time it used to breed, and where does this oviposition takes place also. So this one, It is in captivity. It laid eggs, 7 eggs. Then we found that it has multiple clades. That means after laying eggs, some more eggs are still residing in the uterus. That means earlier, most of the snakes were believed to be breed once in a year. But from here, we found that they can breed 2 or 3 times, more than 1 time in a year. So this is very interesting. So this is their distribution only in Northeast region, Northeast India and the adjacent, like in Bangladesh and Myanmar only. Then this is another paper which we published about all these snakes, about their hatchling behavior, how they hatch, at what temperature, size of this neonate, how long it will take, what kinds of food they take, like that. So that is about Brithia reticulata. Then the second species was we described from Muifang. So we gave the name Brithia Muifang. So now it is no more monotypic genus. So there is 2 species under this genus after the discovery or description of the species. Then on coming to another tree snake, this is cat snake, green cat snake. In Mizo, we call it as rurial. Earlier, local people believe it as a deadly venomous. And the mouth is black in color. But if you look at the teeth, there is no fang. Fang is important for delivering venom. So if they don't possess fang, that means they don't have venom gland. So this is only mildly venomous, not deadly venomous. But it can kill or paralyze rodents, small rodents, and lizards, frogs, like that, on which they prey. And this one is in captivity. Earlier, the maximum record of number of eggs at one time is 10. But now we found that the maximum number is 11. This is in captivity in our lab. This one laid 11 eggs. That is a new record. Then the juvenile is, the body is with brownish color with greenish head. Slowly the color will be transformed into greenish. And this is the tony cat snake. In Mizo, we call it as Rulsaki. This one also, earlier we believe it as deadly venomous because the head is big, more prominent than the body. Then this one is also mildly venomous. And this one, ornate flying snake, Chrysopelia ornata. Earlier we could not find a Mizo name for this. So since the coloration looks like Mizo traditional clothes, this lembuang, so that's why we call it as lembuang rule. It is also non-venomous. This one is one of the most common snakes in the anthropogenic area. It is a copperhead trinket snake. Scientific name is Collignathus radiatus. It is a very good controller for rat population. So scientists, they used to call a good friend of farmers. They control rat population. And another snake that is false-eyed cobra, large-eyed false cobra, because it can mimic cobra by spreading the hood. It is non-venomous also, Pseudoxenodon macrops. And another tree snake, Dendrolaphis cynochloris, Dendrolaphis biloriatus, Dendrolaphis proarchos. All these are non-venomous. And another one, bamboo rat snake, Oreocryptophis porphyraceae. This one is the juvenile, the hatchling one. Slowly, it transforms the coloration like this. Then after it attains maturity, then the color will be like this. So we cannot judge snakes from the species of snakes from their coloration. The most important is scalation. Number of scale will not change, but color can be changed depending on the season and age also. Then this Libeltis toliska, this is the rediscovered species from Mizoram. Earlier, it was believed as extinct in the wild, but recently we found from Mizoram. Libeltis toliska and Libeltis venatus. Lycodon olicas, Lycodon jara, and Lycodon zoy. This one was described from Mizoram, Myanmar, and Assam and Meghalaya in 2001. The one who discovered this species was Professor J.B. Slowinski. This one is a scientist who described Naja mandalaensis from Myanmar. So on that same year, in 2001, he was killed by Craig in Myanmar. He was one of the most promising herpetologists in the world, but at the age of 35 only, he died from snake bite. So this is the what we contribute for him, for his species, this Lycodon zoy. So we studied about its phylogeny, hemipenis, behavior, and how many eggs it can lay, and what kinds of food it ate. This thing we studied in the campus, and it was published in the British Herpetological Journal in 2020. This is the phylogenetic tree for which we construct for this Lycodon zoy. Lycodon fasciatus and Lycodon septentrionalis. This one was spotted in around 1960 in Mizoram. Then after that, after 70 years, more than 70 years, around 70 years, around 60 years, we rediscovered from Mizoram and published in the British Herpetological Journal, the Herpetological Bulletin. And this is Perennia siaboldi. We call it as tuisapai. It is found occasionally in water only. This is the first live photograph from Northeast India. They are very rare snake. And these are kukri snake, Oligodon albosinctas. These are some of our kukri snake, Oligodon dorsalis, Oligodon cinereus, Oligodon cycluras. All of these are non-venomous, but they have very sharp teeth. That their teeth shape is like this Gurkha kukri knife. So if they bite, it can cause bleeding. And this is a rediscovered species, the one which we rediscovered from Tamdil. There is Oligodon catenatus. No specimen can be found in throughout the country. This is the only specimen which is available in our country, Oligodon catenatus, and this one was published in Amphibian and Reptile Conservation, American Herpetological Journal. Earlier it was found in Meghalaya, then that specimen was lost. Then after that we found here in Mizoram. And the type locality is here in northern Myanmar, that is in Kachin Hills. Then another snake, Orthiophis tainuras and Goniosoma prasina. These are also non-venomous. And this is Tyscoros Laiuk. This is also known as Indo-Chinese rat snake. They also control rat population. And Thais mucosa, Indian rat snake. They play a very important role in the paddy fields, and they control rat population and but those which destroy or consume our cereals plantation. So they are also usually known as good friends of farmers, because unlike cats, they can enter the rat hole and kill all the rats family, including the juvenile like that. Cat cannot enter rat hole like that. So that's why they are very important in ecological balance. Another is Rhabdophis himalayanus, Himalayan keelback, and Rhabdophis helleri. This Rhabdophis helleri, which is known as in Mizo, Rul Hongsen, earlier it was identified as Rhabdophis subminiatus. But recently, after studying specimens throughout Southeast Asia and South Asia, it was found that the species which we have in India, it is Rhabdophis helleri. This is the juvenile morph coloration and the adult. And it was found earlier they were they were thought to be non-venomous, but recently it was found that they are also deadly venomous. They don't have poison or venom gland here, but their venom gland is spreading through their gums, and they possess these rear fangs. Once, if they bite and chew for a while, it can create hemorrhage complications, because after biting, there is a groove in their fang through which venom will pour down here, and that will enter in the injured part. And they are also known as poisonous snake, because we should know what is the difference between venom and poison. Venom means those which can create problem through blood. And another one, poison, is those which can create complication through digestive system. So poison and venom is different. Snake, they possess venom, not poison. Okay, because when they bite through blood circulation, it creates complication. And this one, in 2017, I captured this snake in the campus. Then from the neck, from the nostril region, it squeezes out this milky product. This one, if it enter mouth or eye, it can cause blindness. And if it enter mouth and digestive system, it can create complication. So it is having both venom and poison. It is recently discovered. Earlier, it was thought that only this keelback snake, which is found in Japan and Taiwan, is having those kind of behavior, and this poison, it is transformed from their prey, that is toad, before. From the toad, bufotoxin, it is sequestered and stored in this neutral gland. Then that will squeeze out when they are alarmed or captured or disturbed by their predator. So it is a defensive poison. So we studied in our lab about their behavior and natural history. So we made that we allowed them to hatch out and monitor at what temperature and duration, how long it takes, like that, for to make them hatch, under which temperature and humidity. So this one we published in the American Herpetological Journal, Herpetological Review in 2021. Then this one is another recently discovered one, Rhabdophis bildi. If you look at this neutral, there will be sign of bildi, diamond-shaped coloration here. And this one is Himalayanus. You will not find this diamond shape in this case. And on coming to Rhabdops bicolor complex. So earlier these snakes, rain-loving snakes, rule. Earlier taxonomists, they identified it as Rhabdops bicolor. So we collected many specimens, different variety, different morph. After we studied that, we found that they are not only new species, they are new genus. So we gave the name for this snake as in honor of this Malcolm Smith, a British herpetologist who worked lots of these snakes and amphibians from reptiles and amphibians from India. So this is Smithophyes atemporalis because it does not possess temporal scale here. That's why atemporalis, absence of temporal scale. Smithophyes atemporalis. And another one is Xenoprophis piscator. We call it as Imizo, rule. They are also non-venomous, but they can bite severely. And this is their eggs, and this is how we monitor their eggs and hatchling. And this is the same species, but this is leucistic form. Like human being and other mammals, higher animals, because of this genetic disorder, there can be leucistic form, lack of pigmentation. So this one is not a little bit different from albinism. In case of albinism, eye color will be pink. There will be no pigmentation. But in this case of leucistic form, eye color will be dark. It is not pink. So in this way, leucistic form and albinism can be differentiated. So this is the sequence which we generated, and this is this case which is first recorded from this Northeast region. It was published in this Reptiles and Amphibian Journal. And another one, Gongylosoma scriptum. They are very rare snake species, and it was found only in this region, Myanmar, Cambodia, and Thailand. This is the type locality, the place where it was discovered. And about this much, around 1,000 kilometer in aerial distance, we found this snake in Suangpilon and Saita. So this is the first report for the country, new country or a new report for India. It was published in the Hamadryad. Then this one is another snake that looks like this caterpillar, Uitepangang. We call it as Uitepangang rule. It is Eupeplophyes mandarinas. This is the first record from Mizoram. Earlier in India, from only 3 localities it was reported, from Arunachal, Nagaland, then for Mizoram, it is a new record. And on coming to family Peridae, these are snail-eating carriers, snail. They used to consume snail. They are very professional in eating snail. They will coil around and they will take out the body part from the shell. They're very expert for doing this. They are nocturnal. They usually hunt these snails at night time. So we found these two other morphs. This is Parias monticola, which is quite common in Mizoram. And these two, they are very rare species. And after we study their molecular and morphology, we found that this is the Parias modestas and the other one, Parias andersoni, which were already eliminated from the snake species. Then we resurrected these two species, and it was published in Vertebrate Zoology in 2020 with this German, Vietnamese, and Russian scientists. Then family Lamprophili. This is a mock viper, and Samo means soil. That means a soil-loving snake, or lay rule, or rule zekak. We coined the name as rule zekak in Mizo because this sign symbol looks like Sairokherkak, catapult. This is the snake which used to give birth. Maximum 10 neonates. They are also non-venomous, but they can mimic viper. That is why they are known as mock viper. So this is the haplotype and genetic data we generated from Mizoram, and this will be very interesting in future. This one is published in the Zoological Records of India, ZSI journal. Then another family is Xenodarmidi. This is the recently discovered snake, Rule Pusin, Stolischia van Hoelianae. Another family, Elapidae. This is coral snake, the only coral snake which we have in Mizoram. And if you look at carefully, there are three distinct morphs. One with dot on the dorsal body, black dot. But this one with full stripes, transverse stripes on the dorsal. And this one with very less dot. So this one, we studied throughout the South Asia and this Southeast Asia and this China also, where they are found in this Asian region. Depending on more than 600 specimens from different museum. We examined and described two new species from this coral snake, and it was published recently in 2021, along with this Professor Eric Smith from Texas University. This work we started from since 2008. It was very hectic work. We have to examine everything for hemipenis, genetic data, morphology. We need to compare those which are available throughout this their distribution. So this one came out with a very nice paper. And if you look at here in Mizoram, we have these two distinct species, morph. But at present, it is allocated to one species, Cynomicrurus macallandai. But it is very still doubtful. This one, we hope that there will be nice result, which will be coming out very soon. After doing CT scan on their head scale, then body scalation, genetic data, and their natural history. Here, this one, this type, this is also known as Cynomicrurus gorai type, without this full stripe. They used to give birth. This is the first documentation. Luckily, the one which we captured, it laid eggs and they hatch out in our lab. And this one, Cynomicrurus macallandai, it is written as the litter size is up to 7 neonates. But here we found only 3. And moreover, there are many differences in between here. So there might be very interesting thing to come out. Then another elapid snake. Those are, before going to this Cobra, elapid means those venomous snake which are having fixed fang that cannot move. Unlike viper. Viper fangs, it can be it is foldable. But these are, their fang, it is fixed. Like a dog teeth, like a dog, a canine, okay? So their venom is usually neurotoxin. That means they disturb nervous system, okay? So depending on the hood mark also, there are different morphs without any hood mark, with oval shape, with diamond shape, with U shape, like that. But they are same species at present from our study. So this is the one which we studied from Bangladesh and Northeast India about their genetic variation. So this one was published in Mitochondrial DNA Part B in 2020 to show the difference, diversity among these same species in their distribution. This is the phylogenetic data, Najak out here from India, Bangladesh, like that, and their haplotype also. And they used to lay their eggs in the burrow or in the tree trunk hollow. So they used to guard for a while. This one we captured from Triak village. It is in the cultivation area. It disturbed these farmers and it also killed their here. So we published this one in Herpetological Review about our observation. And another crate, that is Bangaras fasciatus, banded crate. In Mizo, we call it as Chonglei or Tiangsir. They are also deadly venomous, necrotoxin. Their mid-dorsal scale, this crate usually with these larger scales with a sharp rich mid-dorsal vertebra, which is hexagonal in shape, this scale. So there is very less study on this biology of this banded crate than this one. We found that they consume other venomous and non-venomous snake. And our observation, it was published in this Data Purbanica from Indonesia last year only. And another crate, which we have in Mizoram, black crate, that is Bangaras niger. This most of the victims from snakebite came from this next to this viper. They came out at night. They are nocturnal. They used to enter human habitation, and when it is very easy to step on them, then in afraid of that, they used to, in return, they used to bite, and they are deadly venomous. So we used to confuse that this is the so-called rulngan. Most of the victim, those patients, then those who lost life from snake bite, are usually from the snake bite, this black crate. So there are also other snake eater, and this is the one which we published about this deadly venomous snake by comparing with the congerer species found in Assam, that is Bangaras lividas. This one, and this is the Bangaras niger, the black crate, and this is the congerer species, which was published in Salamandra, German Journal of Herpetology. This is their distribution. This is in some place they are overlapping sympathetic species, and there is parasympathetical distribution. This red indicates distribution of Bangaras niger. They are found only in this region and highly venomous. And this is Bangaras lividas, which are found towards more western side. So this is the phylogenetic tree, which was the first phylogenetic tree for these snakes, published in the journal. Then when we talk about herpetology, it will come. The first thing that will come to our mind is king cobra. So there was a very long-lasting debate among Mizo community also regarding what is rulngan. There are lots of stories about this rulngan. They used to say it can fly, it can bark, it can pierce through the body like a spear does. The fangs carve out of the mouth. Male and female guard the nest. A male is shiny blackish like that. So after a long struggle, collecting dead specimens, live specimen from zoo, then we examine, then visiting their nest, we confirm that this the so-called rulngan is king cobra, which many people also still don't believe it. Through genetic data, DNA, and morphology, we found that we confirm that this snake is king cobra, because king cobra is the only snake that will nest with dry leaves, if usually in a bamboo area, then guard the eggs like that. So the first description came from in early 19th century, around in 1830, by Cantor, the Danish medical doctor, who was employed by this Eastern Company. So he found them from Calcutta, near Calcutta, then Sundarbans. So he described it as new species, Naja Hana. And later, they changed the name Ophiophagous hana. Ophiophagous means other snake eater. Hana means those ghost, Greek mythology ghost, which usually dwell in trees. So these are also good climber. That's why he gave the name. So their distribution is from Western Ghats to what's Eastern, then till Philippines, Indonesia, Southeast Asia, like that. So their distribution range is very huge also. So there was some curiosity about this snake. It might belong to, many herpetologists believe that it may not be same single species. There might be hidden species. So that's why it is also known as complex species. And it is monotypic genus because under this genus Ophiophagous, only Hana species is available, described so far. So we studied almost around 15 nests of this snake from our state, and we studied about their incubation, size of the juvenile, how many bend throughout the body like the skeleton parts. So if you look at here, the neonate newly hatch out juvenile, the body with transverse pale yellow band, alternate with this black color. Then this one is slowly diminishing. When it gets grow older, then this one sometimes will disappear. Only when it spread the skin or hood, it will appear. So the coloration from olive brown to blackish. This is the olive morph. This is the black morph. Then the one prominent dynastic feature for this species is presence of this occipital scale, true enlarged scale in the nuchal region, in the dorsal head. This one is deformities. And here, this is king cobra, king cobra, king cobra, king cobra, king cobra. This is black ray. In this case, you will not find this occipital scale. This is another king cobra. And regarding their hemipenis also, there is bifurcation here. This is king cobra, hemipenial morphology. This is a copperhead trinket snake, Collineatus radiatus. The hemipenis is different. And this is the coral snake, Cynomicroras macallandai. This is how it appears. And this is the Najacautia, monocrete cobra. So in this way, hemipenis is very important for taxonomy. Many scientists are working on it for identifying species and the marking. Then we compare the genetic data with this banded grade to find out the haplotype and phylogenetic relationship between their phylogeny. So this one was published in Mitochondrial DNA Part B in 2020. And this one, this is the same species. So here we used to say that the king cobra or rungan, they are blackish, but it cannot be said like that because this is the same individual. This was in August 2020. This was in 2021. When winter approach, when the atmospheric air is becoming cooler, as they need more heat. It is very simple. We know that black color absorb more heat. So their body is usually slowly changing into black morph. Okay. This one is still alive in our lab. The largest king cobra which we have ever encountered. And at the same time, this one we published in Sauria, German Herpetological Journal. They also have predator, enemy. This is a juvenile one. Some of our this king cobra baby, they escaped from our custody. So later we found this juvenile cobra which was very plumpy. Then we capture and we keep in the basket. Then after that, it regurgitate this king cobra juvenile. So that means even this juvenile monocoque cobra used to consume or kill this king cobra. So king cobra, though it appears it as top carnivorous, they also have enemy. So this was published in 2020. And on coming to different types of king cobra. If you look at many king cobras on TV, in the Geo or the Discovery Channel, you'll find many different coloration, body shape also. So this is through this species delimitation. This one was published in Molecular Phylogenetics and Evolution with my friends, this Lalremsanga. So there are 4 lineages. Then from this, it can be said that those king cobra in this is our communicated paper that will be coming out soon for describing 3 new king cobras. This one, Ophiophagus kalingana, found in the Western Ghat. And this is the Ophiophagus hana, found in this region. The real king cobra, including Mizoram and other Southeast Asia, Northeast, and some eastern part of India. And the third one is Ophiophagus bungaras, which were found in Indonesia and some parts of Malaysia. And the third one will be O. salvanatana, found in Philippines. So this paper will be coming out soon. It will be a big shock for herpetology. Then another family, Viperidae. Then as I mentioned earlier, these vipers, they have fang that is foldable, elongated fang. Their fang, snake fang, they can be replaceable. Once it is broken, then another new one will come out. Sometimes 2-3 fangs can come out in one this teeth gum. So there are some double or triple fangs. This is the mountain pit viper. This is we call it as rulmuk. Their venom is hemotoxin that can cause tissues and blood. They can destroy blood vessels, tissue, blood also. And this one is the first evidence about this snake behavior, how they lay eggs and guards, and how they juvenile came out from the eggs, under which temperature, humidity, like that. This one was collected from Bualpuyenji. And this is the first observation. It was published in Herpetological Review. And the so-called rule to Ha, Trimeroceras erythuras. Their venom is also hematoxin. This is the most common snake, venomous snake in Mizoram. Many cases are also there. This is the first observation, which the female adult, which we collected from Huifang, it gave birth to 15 neonates. So this is the records, the maximum records for litter rate. It was published in 2020. And dicephalic, this one, and there is curvature in the vertebra also. So this is very uncommon case. And this one was the photograph and the study was conducted from Chipir village. So this is the first evidence for this species, and we publish in Reptiles and Amphibians. And another viper, Zongral, that is Trimeroceras popayaram. This is the male morph. If you look at here, there is stripe, red and white. From the behind the eye, in the cheek, and in the lateral body. This is male, and this is female. In case of female, there will be no stripe, but the eye is red in color. here. If you look at this one, it is yellowish or brownish, but this one, it is reddish. And one of our snake was died in this lab, in our lab, this species. Then when we check the abdomen, we found this kind of parasitic infection that caused intestinal obstruction. So because of this snake died. And when we check the stomach content, we found this helminth parasite. So because of this kind of infection, they can develop this kind of obstruction in their stomach that is fatal to snakes. Then this one, which is we communicated it in journal +1, and this, I hope this will be coming out soon. This is a new viper from Mizoram Meghalaya. If you look at the stripe, it looks very similar with Pop Speed viper, but from the eye, it's not reddish. This is the hemipenis, how the hemipenis looks like. And skeletal structure, and this is the phylogeny. If you look at here, this is the tree. This clade, the new one it is quite different from the Conjera species like Medowensis, Unionensis, like Gumpresti also. So many things will be coming up from this work. So it is estimated that between 35,000 and 50,000 people die of snake each year among India's population. So if you look at this fatality rate, it is very high in compared to other disease. So this is the snake venom, how it works. From here, venom gland to this venom duct, it will pass out to the venom canal in the fang. So from this fang structure, this syringe or injection syringe needle is developed by this Greek scientist in around 10,000 BC. So this is how we deliver venom to the victim. This is the front general capturing teeth. So this is the rear fang in case of redneck keelback, Rule Hongsen. So rear fang teeth with venomous flow, it's mildly venomous and venom content is hematoxic. But now it is found that it is not mildly, it is deadly venomous. So we should be careful about this snake. So snake bite symptoms, it is very complicated. From central nervous system, dizziness, fainting, increased thirst, headache, vision blur, heart vessels, muscles, gastric, intestinal, skin. Then wound side also, it can be burning sensation of swelling. There are so many complications involving in snake's bite. So we should know which snake is having venom and what type of venom, whether it is neurotoxin or hematoxin. Snake venom, it is broadly classified into two types, neurotoxin and hematoxin. Neurotoxin, it is possessed by these vipers, then hematoxin, elepid, like cobra, coral snake, and crates. This is the when snake bite happens, do's and don'ts. Take the patient to, which we should not do is not to take the patient to tantric or snake charmer or for treatment. This is the earlier belief, which is very dangerous. Suck the wound, cut the wound open, tie ligature around the wound, burn the wound, apply harbor paste. Because of this thing, we lost many lives. Okay, so this is what we have to do to immobilize the affected limb with supporter. Apply basic first aid, wash the wound with soap and water. But nowadays the WHO don't suggest washing with soap, because when washing, we used to scrub the skin, the wound part. If we scrub and make a friction, then it will faster the blood pressure to transport venom more rapidly through the circulation. The best is to rush to the hospital. Suppose if it is in the jungle, if someone is bitten by snake, this is the way how to do. If we don't have this bandage, we can tear off clothes, t-shirt or gamcha or towel or like that. Then with the help of this supporter, we have to wrap around the wound with not too tight, because tornic tight binding into the or pressure to the wound part is not advisable, because it will create blood pressure more faster. So it should be the wound part, body part should be relaxed as possible as it is. So if it is necessary, the patient should be carried by his or her friends to the hospital. This is the snakebite case reported from civil hospital. If they are treated with medicine, okay, properly, they will survive. No need to treat with antivenom also for this viper. This one, it happens in 2011, 2010. A viper bite from Serchip town. He was died because he went for fishing in the stream. Then he got bitten on the toe by this small viper, Rultuha. Then after he went home, then his friends, they said, you take alcohol, it is very good. So he took alcohol, he drank alcohol. Then at night he fell unconscious, and he could not wake up. So his family and friends, they took him to Aizawl hospital. So they called me up, and then I asked about the situation. Then he died before reaching the hospital. So when they examined his vein in the brain, it was punctured because of that snake venom. Because when he drank alcohol, then alcohol increased heart rate. So that venom on the leg part, on his foot part, came to the lungs, then to the heart, then from the heart to the brain section. So that caused puncturing of the vein. So because of that, he died. So this drinking alcohol is very dangerous after snake bite. So that's why you should know about what is snake venom. It is only protein, toxic protein. Snake venoms are secretion of venomous snake, which are synthesized and stored in the venomous gland. The glands secrete the zootoxin in a modification of the parotid salivary gland and are situated on its side of the head, below and behind the eye, encapsulated in muscular sheath. The glands have large alveoli in which venom is stored before being conveyed by the duct to the tubular fangs, through which it is injected. Their venom gland, it is a modification of salivary gland, which we have in our mouth. So in their venom gland, they store this venom. It is a combination of many different proteins, peptides, and enzymes, and they are generally not dangerous when ingested. Because if we drink venom, if there is no injury in your digestive system, you will not die. Because snake venom, it is protein. Once if you swallow or if you drink, it will be destroyed by digestive system, our protease enzyme. So it cannot function. Okay, it will be digested. Snake venoms are complex mixture of enzymatic and toxic proteins, which include phospholipase A2, myotoxins, hemorrhagic metalloproteinases, and other proteolytic enzymes, coagulant components, cardiotoxins, cytotoxins, and neurotoxins. composition of venom. So snake venom, they contain more than 20 different constituents. See, more than 20 venoms, protein toxins, like procoagulant enzymes, hemorrhagins, cytolytic and necrotic toxin, hemolytic, myelolytic, phospholipase A2, presynaptic neurotoxin, found in Elepidi and some Viperidae, postsynaptic neurotoxins. So it is very complicated. So there is no cure for this snake venom from medicinal extraction. This medicine from the bioactive compounds from medicinal extraction, they may neutralize one or two toxins of snake venom, but we should think about others which are also fatal. So there are the only cure for snake bite is to treat with antivenom. Okay, in the case of usually neurotoxin. Composition of snake venom, it consists of protein, enzymes, neurotoxins, coagulants, anticoagulants, and substances with cytotoxic effect. It has acidic pH. Specific gravity is 1.03. So this is some properties of snake venom. Antivenom. On coming to antivenom, the only available treatment against snakebite is the usage of antivenom. The first antivenom was developed by Albert Kalmit against the Indian cobra Naja Naja. Antivenom is made by immunizing mammals such as horse, goat, rabbit with particular snake venom, and the specific immunoglobulins are isolated from the blood. So it is classified into monovalent. That means antivenom which is prepared from only one type of venom. That means venom taken from one species. Another is polyvalent, when they are effective against a range of species. Polyvalent venom prepared from venoms from different species. As I already mentioned there, big four snakes. From their big four snakes, they prepare their polyvalent. Two hematoxic snake, Naja Naja and common crate. And another two neurotoxin, Naja Naja and common crate. Then hematoxin from two vipers, Razel viper and Soskel pit viper. But we don't know whether that antivenom will be effective to the bites of snakes from our region, because there are more than 20 deadly venomous snake species in our northeastern region. So this is how snake venom, antivenom is prepared. From the snake, venom was collected, then inject with very small dose, very low dose, they injected to the animal. Then after inject some these B cells of this horse, it will develop antibodies. So the blood serum will be drawn that contains antibodies. So it is separated and antibodies is in the concentration. Then this one, it is used for combating venom. So this is known as antivenom. The venom, which acts as antigen, will react with the immune system of the animal, then which in turn will produce antibodies that can kill, neutralize venom. So that is utilized for antivenom. So that means antibodies will contain all the necessary neutralizing factors for different constituents of venoms. Okay, so that's why it is complete medicine for snake bites, venomous snake bites. But in the case of extraction from other, like sodium bicarbonate injection, tamarind dust, and medicinal plants, They don't possess all the necessary factors that can neutralize the constituents of venom. That's why antivenom is the only medicine for curing snake bites. So this is how neurotoxin works. This is, as we know that our body movement, everything, it is under the will of nervous system. Our nervous system and muscles, they are interconnected. And message is sent through these neurons, through axons, then from the presynaptic membrane to postsynaptic membrane. This is the muscles. This one, if it sends signal that is in the form of neurotransmitter, that will attach on the receptors, which is specific. Only this neurotransmitter can bind to its own receptor. Other neurotransmitter will not be able to bind on it. It is very specific. So after it binds, it will activate to contract muscles or to relax. So our movement is under our control of nervous system. So this is how neuromuscular junction function. But in the case of snake venom, this venom, it can mimic this neurotransmitter. It can also bind on the receptors. Then it will bind. Okay? So it will block the channel. The neurotransmitter will not be able to stimulate the muscles or other post neurons. So there will be paralysis catastrophically. Then there will be disturbance in the sodium potassium ion exchange. Usually these create cobra, king cobra bites, which are neurotoxin. They are used the most prominent symptom is respiratory failure, because our lungs, it has no muscles to make it contract for exhalation and relaxation to inhale air. Okay, so it is under the control of the surrounding muscles, that is diaphragm, which is present in our chest region. So that once when it contracts, it squeezes out the air from the lungs. Then when it relaxes, it will inhale oxygen from the air. Okay, so this movement, diaphragm movement, is under the control of neurons. So once if snake poison venom reach that region, then the diaphragm is not under cannot be controlled by these neurons, because the neurotransmitter that will control the diaphragm will not be able to bind on the receptor, because the snake venom will bind on it. So that's why there is difficulty in respiration. So usually patient died from respiratory failure. Yeah, if there is no ventilators like that. But in the case when we applied antivenom, that antivenom will neutralize the function of this snake venom. It will bind on it, and this venom molecule, it will not be able to bind on the receptor, neurotransmitter receptor here. So it will be free for neurotransmitter that is secreted from the neuron. I hope it is clear. So the fact in our country is like this: 50,000 people die every year in India due to snake bite. Most of the victims are poor village dwellers, farmers, laborers, children. Snake bite treatment in India is still in the trial and error method level. More than 70% of India's population are occupationally vulnerable to snake bite because they are living in rural areas. Most of them are sleeping in the floor, on the ground, not on the bed, which are more vulnerable to snake bite. ASV antivenom, okay, is still not as popular as condoms in India. If you think about how many people used to die because of HIV in India, only very few if you compare with snake bite. a large section of Indian population are still dependent on the traditional method of snake bite remedy, like traditional belief. Okay. In case of Mizoram, if someone is bitten by snake, if they call me, then I used to ask, what kinds of medicine you applied first? Then they usually say, Chang El Tui, I drink this banana fruit, banana juice, Musa paradisica, Musa sylvestris juice, like that. And Vakep gives Muswendra Gabra. So because of that, they used to delay their time. That's why many in the rural area used to lose their life because of wrong medication. So this is which we should not practice again and again. What do we visualize when we say snake bite in India? The snake bite dead corpse is then placed on a raft and set sailing in a river or sea. The belief is that they might come back. This is 21st century India. Mind it, we are in the computer age. And another funny thing is that some people, they used to sell false fake medicine. They used to carry non-venomous snake like this copperhead tinker snake. So they tested them, but these local people, they are innocent. They thought that this is venomous, so they applied that medicine. So there nothing happened on that victim. So they thought that that medicine works. So that's why they used to sell that medicine is very costly, which is very costly. Even if they don't apply that medicine, he will survive. There will be no symptoms at all. So in that way, some people, some culprit, they used to cheat local people. So these are some of the plants used for snake bites, which are from which active compounds were extracted in different research institute. But as I mentioned earlier, all these active compounds from these plants, they neutralize only one or two toxins out of those more than 20 constituents of venom. Traditional approach to treatment, ASV, antivenom, antiserum, is an unknown substance, no relevant education in the front line. Front line, those people who work for us, who do all this labor work for cultivation, who are in the field. That's why they are front line. They are prone to different risk of life, like malaria, snake bites, like that. This child died 28 hours after being bitten by a naja, no hospitalization, because they are very innocent, they are illiterate. They have no idea about snake bite with antivenom. So this is how what happened. Story of our country. This is the story of four stakeholders of our country. Name everybody, somebody, anybody, and nobody. There was an important job to be done, and everybody was sure that somebody will do it. Anybody could have done it, but nobody did it. Somebody got angry, and it was everybody's job. Everybody thought that anybody could do it, but nobody realized that everybody could not do it. It ended up that everybody blames somebody when actually nobody accounts anybody. So this is the hidden deaths. Most deaths are occurring outside in the hospital setting and in rural areas where they are often not registered. The MTS is starting to expose the hidden trends. Cancer day, see here. Malaria. This is inside hospital. This is in the rural outside, okay, where there is no hospital. Suicide, road traffic injury, HIV, snake bite. If you look at, it is highest in the case of rural, as well as in the second highest in the hospital. So it is a major public health problem. yet the union, this is the previous former Union Health and Family Welfare Minister, Mr. Ghulam Nabi Azhar, informed the Lok Sabha in April 2012 that there had been a total of 1,440 deaths due to snake bites in India in 2011. He added as many as 380 of these cases from West Bengal, our neighboring state. Why? The key reason why snake bite is not a priority is that most victims lack political voice because they tend to be poor children and from rural areas. So this is the saw-scalped viper, one of the big four snake, Bangara cyrillus, common crate, Naja Naja. These are not available, though they are not available in our region, but we are living with the many other species which are equally significant. Okay, as I mentioned earlier, more than 20 deadly venomous snakes are distributing in the northeast region, and against whom ASV antivenom, okay, might not be effective. For example, these are the snakes which I had already shown: king cobra, banded crate, black crate, banded. So So what do these venoms do? This is the hematoxic snake bite, which can disturb or destroy blood tissues like that. The pain, it causes pain and slow swellings, rapid, sometimes ignored, bleeding from any side, clotting disorder, late hematuria, oliguria, acute renal failure. Then, white blood cell test is also very important. After 20 minutes, whether the blood can be clotted or not. In the case of this viper bite, then another hematoxic bite signs, viper bites, okay, like rule tuha, rule muk, vomiting and fainting attacks, pain abdomen, hematoma at injection site, then the regional lymph nodes. So immediate tissue necrosis in cobra bite, compartment syndrome in viper bite, gangrene due to tight ligature. This is because of the ligature. Venom will be concentrated and sometimes it needs to cut it off, okay, due to this gangrene development. Last year also, there are lots of this kind of case reported to us because of mismedication. Cobra neurotoxicity, pain and swelling, rapidly develops paralysis, bilateral ptosis, ophthalmoplegia, blurred visioning, slurring of speech because of swelling of tongue, nasal inundation, dribbling of saliva, difficulty in swallowing, respiratory distress, respiratory failure, great neurotoxicity. This neurotoxicity from great cobra, king cobra, and coral snake. The bite is usually painless, no local sign, pain abdomen, pain in throat, pain in joints, very slow developing bilateral toxicity. That's why people used to neglect. Difficulty in swallowing, slurred speech, tightness in chest, paradoxical respiration, respiratory failure. Usually this happens at night time. Then people used to ignore it. Then usually they lost their life in the early morning like that. Very easy to neglect this great bite. This is so. The principles of management of snake bite is to retard absorption of venom into circulation. So we have to avoid of snake venom to enter more deeply in the circulation. As we know that our blood circulation is not so slow. Once if you inject it, then it will disperse or distribute it to the deeper part of the body. So in this way, by wrapping with this bandage along with the supporter, it will reduce, and not to keep to avoid movement of that wound part. Okay, if it is keep on moving and moving, then there will be more increase in the blood circulation to neutralize absorbed and circulating venom. To treat manifestation of organ dysfunction caused by venom fixed to the target receptors, to arrange transfer to upper level care, set up where necessary, recording and transmitting data of outcome. Field management is important. Level 1 is PHC subcenter kanenchi hotemo. Level 2 SDH HDU. Level 3 care apex institution with ITU and multidisciplinary health care setup. Tools for management of snake bite test. 20-minute whole blood clotting test. This is very easy to conduct in the laboratory. We need clean and dry glass test tube, 2 ml of blood of snake and victim. Result interpreted at dot 20 minutes. After 20 minutes, we can observe it again, whether blood is clogged or not clogged. If it is not clogged, that means it is because of viper venom. Level 1 care tools, anti-snake venom, neostigmine, atropine. These are all basic medicine which must be available in the casualty or PHC or hospital. Level 2 care tools, all level 1 care tools, observation protocols are snake bite can be also dry bite, because if it bites, snake usually restore or reserve or conserve their venom for their prey, for their food, not for their enemy. That's why when snake bites also, they may not inject their venom. That is known as dry bite. Then we should be able to identify from the symptoms whether it is neurotoxin bite or hematoxic bite. Okay. In case of neurotoxic, there will be very less sign, blur visioning, protruding of the tongue, respiratory failure, difficulty like that, vision problem like that. But in the case of hematoxic, the wound part will be swollen, bleeding, gangrene like that. So in that way, it can be differentiated easily. Level 3 care tools. These are set up in the hospital, okay? Like ITU, dialysis, hematology, surgery unit, follow-up OPD setup, research unit, and MRD. Mistakes we continue to make, we become very apprehensive of treating a snake bite patient. We panic. This thing we should not do like that, okay? If we panic, heartbeat rate will be increasing. That will increase circulation of venom into deeper part of body. Try to shake off the responsibility by referring the patient to some other hospital in the name of better management. Scared of using ASVS, reluctance and incompetence in intubating gasping patient, prescribing tests which do not have any relevance to the then snake bite patient, not attending the snake bite patient continuously before and after ASVS administration, not learning about the snakes in the catchment area. Then this thing we should not forget. People who die due to snake bite are the ones who produce crops for us. People who die due to snake bite are the ones who build our house, roads, railways, canals. People who die due to snake bite are the ones who carry our loads. People who die due to the snake bite are the ones who make this nation self-sustained. People who die due to snake bite or are our fellow countrymen and women. People who die due to snake bite are our children. People who die due to snake bite are, after all, human beings with dreams. Then at the same time, we should know about their role in the ecosystem, the snake. Food chain among snakes and other animals which help a good ecological balance. Cat snake, rat snake, trinket snake feeds on rat and controls the rat population. Then banded crate, monoclade cobra controls other snake population and also rat mice. King cobra feeds on banded crate, monoclade snake and other snakes. also feed on rat, mice, and thus helps in balancing the other snakes and rodents population. So this is the food chain concept. We are interlocking. All organisms are interlocking in this food chain and food web complex. So this is the king cobra. If you look at this as their food chain, if we express in the pyramid, food chain pyramid, food grains which are required to human are eaten by rats and mice. About 2/5 of our cereals and crops are eaten by these rats, mice, and birds every year. Okay, if snakes are not there, then what will be happen? Whole country will be under famine condition. Cat snakes, rat snake, tinket snake controls mice, rat, amphibians population by eating them. Banded crate, black crate. So this population, increasing population is under the control of another snake, another venomous snake. Then another snake is controlling this deadly venomous snake, which is king cobra. Earlier, about 2-3 years back, in Mizoram also, we killed so many king cobra. Whenever we encounter king snake, we used to kill. Then later on the next year, these black crate, their population are increasing like anything. That's why many snake bite cases happened during that 2-3 years back. So this thing, which you should not do, the best is snake repellents. Repellents, which are keep on developing in the commercial. And this thing we should not do, not to handle snake, which we do not know, better to chase it. Or with the help of snake wrangler. So killing of snakes also can cause or disturb ecological balance, as we had seen, like other organisms. We are interlocking in the food web. Which organism is important for others? We have to care for our planet. Though I really want to campaign for plastic management, but there is no such time. So we have to care for our planet as there is no planet B. So this is the myths about snakes. Snakes attack human beings. Then the fact is, snakes attack only when humans accidentally step on them or when they get the feeling that they are attacked by human beings. Snakes drink milk, according to the myth, but in the facts, snakes hate milk, and it is scientifically proved that when snakes are forced to drink milk, they get sick. And another myth, snakes listen and move their hood as per the music by a charmer. Then the fact is that snakes cannot hear music. It just follows the movement of the snake charmer, which forces the snakes to move their hood in a particular movement. Snakes, when hurt, would definitely come back to take revenge, according to myth. According to the fact, it is scientifically proved that the brain of a snake is not much developed, therefore it has absolutely no memory to take revenge. Then another myth is that if a snake bit a person in turn bites the snake, the poison would go away from the body of the victim. This is very funny. And the fact is that if the victim takes the risk of biting the snake would only attack the victim again, and this would only result in the snake injecting all its poison into the body of the victim, which would in turn result in quick death. Then another myth, snakes have supernatural powers, and a golden color snake has diamond in its mouth. Then the fact is, snakes are ordinary creatures just like other animals or other reptiles and have no supernatural powers. Snakes do not have diamonds in their mouth. It is just a fantasy and a story for movies. With this, thank you.
Sir, thank you for your insightful presentation in enlightening us with herpetology, sharing your valuable knowledge and the do's and don'ts which are really applicable, relevant for all of us in our daily lives. We are really fortunate to have you tonight in our midst.
Thank you.
If there are any queries or questions among the participants, I kindly request to unmute yourselves and ask the resource person directly. Queries? Please feel free to ask in Mizo language. Okay, I think it's clear enough, sir. So once again, we are really thankful for accepting our humble request. Your contribution towards Mizo society is highly dignified, and Government Certificate College wishes you the best of luck for your future endeavor. So with that, we have come to the end of tonight's program. Thank you all for all the participants, and please do join us tomorrow night at the same time, 8 p.m. Stay safe. Good night, everyone.
I hope all of you are still awake. Okay.
Yes, sir. Yes, sir.
Sharing with
We're
me.
very much awake, sir.
Cha mai lo ngai thak tak mo.
Ngay thải laptop liền to kin.
Angai thak tlak em.
Ngay thak ke.
Saraman lalu tu kan ya kan log pulu tu.
Kan long khóc này.
Anh mang thả tháo à?
Vâng thở lên.
Traffic clear.
Sale.